All right, so hello everyone. Thank you for joining this talk, especially in the midst of all of our crazy power outages across the nation. So just like to introduce myself. My name is Frederica Lamar, and I'm currently a fourth-year doctoral candidate in the Environmental Health Sciences program at Emory University. And today I will introduce the Cheap Cheap project and results from our first phase of study. So our work suggests a food systems approach utilizing microbial measures to assess key hazards and risks to child health associated with chicken-related enteropathogens. As some background that informed the study, as we all know, WASH studies have routinely focused on removing human waste. However, along the modified F diagram, there are both primary and secondary pathways that expose humans to animal feces. So Delahoy et al. 2018 found that 28% of diarrheal deaths in children less than 5 years old were attributable to 4 pathogens, and all of these pathogens are transmitted via animal feces and found in poultry. Animal husbandry and the production of animal feces is increasing worldwide, and organizations are promoting small-scale and family poultry as a development strategy to provide nutrition and income. So given the promotion of small-scale and family poultry and the burden of diarrheal disease due to pathogens carried by poultry, our work provides a mixed methods approach to first understand food safety hazards and risks along an animal value chain before then developing mitigation strategies. So our project is in Maputo, Mozambique, where there is a high prominence of promoting and selling poultry. Our pathogens of concern are Campylobacter and Salmonella, and the purpose of this work is to potentially identify intervention strategies to mitigate risks associated with these exposures. So our project has four main research questions. The first is, what are the locations and behaviors that expose children to chickens and chicken feces? Second, to know where along this chicken value chain these pathogens are found in the highest concentrations. Third, to generalize the chicken value chain across contexts. And lastly, to develop potential strategies to address these high-risk exposures. So in 2018, we completed formative research to map the chicken value chain and assess potential childhood exposure to enteropathogens from chicken fecal contamination. We did this by performing informal interviews with small-scale and commercial farmers, feed companies, depots, which sell day-old broiler chicks, live informal market vendors, grocery store clerks, veterinarians, and local poultry foundations. Based on these interviews, we identified key settings for sampling in our first phase of work, and we mapped preliminary value chains for broilers, which are your meat-producing chickens, layers, which are your egg-producing chickens, and indigenous chickens, which are free-roaming chickens producing both meat and eggs. And then using this information, we outlined the first phase of our work. So in 2019, we went back to refine our value chain maps, and we quantified and characterized contamination at key settings along the chicken value chain. And today's talk will focus on the results from this first phase of work. So in 6 of 7 Maputo city districts, we conducted semi-structured interviews and collected pulled chicken feces and carcass risk samples at 6 key settings. And we identified these settings based on our formative research. We conducted a total of 78 semi-structured interviews across 68 sites. So this first table is just laying out the settings per district and the number of interviews. And the second table is summarizing our respondent characteristics. We also conducted observations at 8 markets to better understand food hygiene practices and any potential additional exposures to children. A total of 193 samples were collected, including 118 pooled fecal samples and 75 whole carcass rinses from broilers, layers, and indigenous chickens. So this table is showing our sampling scheme for those pooled fecal samples and carcass rinse samples. So for chicken feces, we collected 4 separate fresh fecal samples and placed on ice. We then pooled equal amounts to total 1 gram of sample and extracted using the QIAGEN Dionisi Power Soil kits. For the carcass rinses, we collected freshly butchered chickens and whole frozen broilers and placed them into large poultry rinse bags and then on ice. We rinsed them in buffer peptone water solution and then extracted them as well. And we ran assays on these samples for general salmonella and Campylobacter jejuni and coli. So I'll first discuss our qualitative results. So as I said, we mapped value chains for broilers, layers, and indigenous chickens. And I'll now walk you through the value chain for the broilers. So for broilers, breeder chicks are imported from neighboring countries and produce day-old chicks for meat production. Chickens are either commercially or informally produced, and commercial chickens are marketed to grocery stores and restaurants to the consumer. Commercial producers may outsource production to medium-scale farmers, also known as outgrowers, who receive farming inputs from commercial companies, raise the chickens, and then return them to abattoirs for processing. Broilers are also produced by small-scale farmers who buy day-old chicks from commercial depots. These chickens are then sold to smaller grocery stores and restaurants, to informal market vendors who then sell directly to the consumer or to neighbors and surrounding households, or informal market vendors resell chicken parts to corner stores who then sell directly to the consumer. Also, frozen whole chickens and parts are imported from neighboring countries and sold at grocery stores, restaurants, and corner stores, or are purchased directly by the consumer. So here, the commercial and informal sectors play an important role in chicken production, with the informal market functioning as a key node connecting those sectors. Here's a layer value chain. Similarly, commercial and small-scale farms produce layers and eggs and market them similar to the broiler value chain. However, most of egg production is still external to Mozambique. Lastly, the indigenous chicken production is the simplest of them all. So chickens free roam in the community and are either gifted or sold at markets. And I will now go through our prevalence data for each pathogen tested. So for our pooled fecal samples, general salmonella was detected in 11% of samples, and Campylobacter jejuni, which going forward I will just call jejuni, was detected in 71% of samples. And the sample numbers are denoted above each bar. At markets, feces from each type of chicken tested positive for both salmonella and jejuni. And indigenous chickens from households tested positive for both salmonella and jejuni. For carcass rinses, overall, general salmonella was detected in 21% of samples, and jejuni was detected in 69% of samples. And like with the pooled fecal samples at live and formal markets, carcasses from each type of chicken tested positive for salmonella and jejuni. And most notably, 100%, so all of our carcass rinses collected from informal markets were positive for jejuni. Now I present data on the concentrations of salmonella and jejuni in our samples. So please also note that this data is presented on two different scales due to differences in the order of magnitude between salmonella and jejuni. So for pooled fecal samples, market broilers were most concentrated with salmonella and Campylobacter, and concentrations of Campylobacter were 3 orders of magnitude higher than those of salmonella. And then for carcass rinses, the grocery store broilers had the highest concentrations of salmonella, and indigenous chickens at markets had the highest concentrations of jejuni. And concentrations of jejuni are an order of magnitude higher than that of salmonella. So through our interviews and observations, we also gather additional data to characterize potential exposures to children. So at depots, children were observed but had no contact with chickens. At grocery stores, no children were observed. At corner stores, children were observed but had no contact with chicken products. And of the 27 farmers, 41% of them had children who lived at the household. Of the 20 market vendors, 25% of them reported children playing with or touching chickens, and children were observed purchasing chickens and eggs or with their parents. And of the 19 household respondents, 63% of households corralled chickens inside the home at night, 32% of households reported children helping with butchering activities, and chickens roamed in child play areas at 63% of the households. So now pulling this all together, pulling together our quantitative and qualitative data, we return back to our value chain maps to highlight key settings with potential high-risk exposures. So icons in grayscale are settings with potential pathogen accumulation, and child icons are next to settings with potential exposure to children. So for broilers, those settings are households, small-scale farms, and informal markets. And for layers, the same settings are highlighted. For indigenous chickens, key settings are households, informal markets, and the greater community where chickens free roam. So there are some important takeaways that we gathered from this first phase. First, informal markets are a key node of risk that have been largely ignored. We saw a clear overlap between the commercial and informal sectors. All of our chicken carcass rinses were contaminated with jejuni, and both pathogens were detected in each type of chicken. Commercial chickens were also contaminated, so we can't make the assumption that just because chickens are commercially produced, that they're necessarily safer to consume. And we saw that management practices and sanitary risks introduce opportunities for potential pathogen accumulation. And bringing it all together, the value chain framework can be implemented as a tool across contexts to first understand food safety hazards and risks along these value chains before developing interventions. Our work highlights the need for a greater focus on food-related pathogens, LMICs, and it represents a collaboration between WASH and veterinary sciences. Our work also lays the foundation for future work to further assess risks. In learning from our data, we are currently executing our second phase of data collection. So we will quantify consumption and the flow of chicken products along these value chains and investigate contamination at high-risk settings identified from our first phase of work. So we plan to further study pathogen accumulation at live and formal markets, mainly focusing on that butchering component, get population-based estimates of the relative consumption and sources, and do additional microbial sampling at key places identified from our first phase of work. So I'd like to thank everyone, our funder, our research team, our in-country collaborators from Eduardo Mondlane University, Maputo City Municipality, and all of our study participants for making this work possible. Thank you for listening to my presentation, and I'll take any questions that you may have.
Thank you so much, Frederica. I really appreciate your presentation. That was great. For anyone listening in, go ahead and post any questions you might have in the Pathable chat. Again, not the Zoom chat, as that is disabled. I'm going to give a couple minutes. In the meantime, I actually have a question for you. So you mentioned kind of the collaboration between WASH and veterinary sciences. Could you speak a little bit more about why that's such a critical kind of combination and why that collaboration is helpful?
Of course. So in the past, WASH interventions have focused mainly on the elimination of human waste and human sanitation, but especially in low resource settings where you're encouraging the production of livestock as a means of nutrition and subsistence. We haven't really paid much attention to the role that animals play in contaminating domestic settings. And when we do look at domestic settings, we sometimes overlook settings outside the household where children could have potential exposure to these enteropathogens. So by working with veterinary scientists and having WASH scientists collaborate, you get those perspectives from each side. So we understand more the human component, and the veterinary scientists understand the carriage of pathogens in livestock and what those overlaps may be.
Great, thank you. Last chance if anyone wants to ask a question in the chat. I'll give you a few more seconds. Okay. Oh, there's one. Last second. So what are trends in poultry production in Mozambique, such as commercialization or things like that?
Yeah, so poultry production is increasing a lot and very rapidly. So right now, egg production, so your table egg production is still mostly external, but it is increasing, and there are government programs to further push the development of small scale poultry. So right now there is an upward trend.
Thank you. And we've had a couple more questions come in. We only, I think, we have time for two. From your qualitative data collection, can you say anything about whether hygiene measures were taken by households and consumers were adequate to mitigate pathogen exposure where you found it?
Yeah, so we did not assess whether or not the hygiene measures were adequate as far as if a household, for example, has a closed off child play area, how does that influence exposures to chickens? So we know that that's ongoing work by other researchers. But for this first phase of work, we mainly just focused on our chickens present at the household, our children present at the household, and what are those basic interactions. And also during our second phase of work, we would do household observations to further assess those interactions.
Thanks. And we have one more question. It says, what kind of sanitation efforts do you think would be feasible to implement at the informal market or small farm setting?
Yeah, so we're super interested in the informal market setting. So for the sake of time, I'll just focus on that one for now. So we're actually going to study the potential accumulation of pathogens in butcher water. So the main issue at the markets is the availability of fresh water to butcher throughout the day. So unfortunately, a lot of vendors by midday are having to butcher reusing the same water. But I know water availability is a very complex issue. So maybe either doing something around that area or some way to sanitize the water as you're butchering chicken. So something in that area.
Thank you so much for answering all those questions and for a
Thank
great
you, everyone.
We're going to move on to our second speaker and final speaker, Gabrielle String from Tufts University. Gabrielle, if you want to go ahead and share your screen, please.
Okay. Is that working? Perfect. All right, so good afternoon, everyone. I'm going to present to you today on a laboratory study that we ran evaluating the efficacy of chlorine disinfection by 2 different application methods, spraying and wiping against Vibrio cholera on surfaces. So a little bit about cholera transmission. We know that cholera traditionally affects those who are lacking adequate wash services. And it's been estimated that approximately 1.3 billion people live in endemic countries and are at risk for cholera infection. Traditionally considered a person-to-person transmission, there's new evidence that environment-to-person transmission via fomites is possible. And we also know that individuals who are living less than 50 meters from a cholera index case are up to 56 times more likely to contract cholera. So that means that surface disinfection is going to be really important in these contexts. And in the household, there are 2 different approaches for carrying out disinfection. The first is via household spraying. This has been traditionally implemented in many contexts. An agent will go into a household, such as the agent on the left, carrying a sprayer that contains a chlorine solution and spray that in different areas of the household, sometimes targeting where spills may have been, latrine areas, sometimes kitchen items. But currently, international guidelines are deprioritizing or explicitly do not recommend the implementation of household spraying. And there are many reasons for that. I've highlighted a couple here. A primary reason for not recommending household spraying is that there's a lack of evidence that it's efficacious and that it can be effective. There are a lot of concerns that it's quite stigmatizing to the household that is receiving the spraying. Household spraying occurs once, and there's the possibility for surface recontamination within the household, and it also can be resource and staff intensive. So a newer model for household disinfection that's been promoted is the distribution of household disinfection kits. There has been limited implementation of disinfection kits to date, and also a lack of evidence on their efficacy and effectiveness. And there's also a lack of standardization of what goes into a household disinfection kit. So these are different than a hygiene kit, which you might be familiar with being implemented in emergency outbreak response. But what really denotes a household disinfection kit is that they contain materials that a household would use to disinfect surfaces, such as cloths and sponges, soaps, and chlorine solutions. And currently, international guidelines do recommend the distribution of these, and particularly highlighting that a household could repeat disinfection by using them more than once. And then surface disinfection is also important in healthcare facilities. Guidelines state that there can be spraying or wiping of chlorine on healthcare surfaces and recommend 0.2% solutions for when the contamination risk is low, and a 2% solution when the contamination risk is high, such as around a fecal spill. Now, while guidelines specifically call out the use of spraying in these contexts, wiping is not as specified, but it would be a similar application as outlined here for spraying with 0.2% and 2% in different areas. But we know from other healthcare facility disinfection literature that the method of applying chlorine to a surface when you're wiping chlorine across the surface is really important, whether you use a pre-wetted towelette, whether you place it on the surface first and use a dry towelette to wipe it off afterwards, and that the wiping pressure can vary from person to person and have an influence on the effectiveness. So what we did in the laboratory was to evaluate the efficacy of spraying and wiping guidelines against Vibrio cholera on different types of surfaces. And you can see from the images below some of the surfaces that we had inoculated and then disinfected. So our testing matrix generally looked like this. So first we were focusing on 10 different surfaces. We wanted to move beyond looking just at stainless steels and plastics, which are more common to some healthcare facility settings, and try disinfecting surfaces that might be more common to low resource contexts and households, including unpainted wood, cloth, foam, and dirt. We applied a chlorine concentration of either 0.2% or 2% to the surface. We tested chlorines made from 3 different compounds, so sodium hypochlorite, high test hypochlorite, HTH, and sodium dichloroisocyanurate, NADCC. The exposure time was either 1 minute or 10 minutes of the surface to the chlorine before it was neutralized with sodium thiosulfate. And we applied by either spraying the chlorine to the surface or wiping a pre-wetted towelette across the surface. The surfaces were inoculated with 2 milliliters of Vibrio cholera at a concentration of approximately 10 to the 6th CFU. And the concentration of the chlorines was always confirmed to be within 10% of our target concentrations. We tested our entire matrix in duplicate with positive and negative controls for 240 total tests. Looking at our results first for spraying. So what we have here is on the x-axis, each of our surfaces organized from the least porous or the non-porous surfaces being the stainless steel, the whole way down to our most porous being the dirt. And on our y-axis, we have our median log reduction values. So when we sprayed 0.2% chlorine and left it in contact for 1 minute, we saw that 7 out of 10 of our test combination surfaces, we achieved greater than 3 LRV. When we then let that chlorine sit on the surface for 10 minutes, we had 9 out of 10 surface test combinations that achieved greater than 3 LRV disinfection. And when we looked at applying 2% instead of 0.2% concentration, we saw that we had even higher number of surfaces that achieved greater than 3 LRV. And I'll note that at 2% and 10-minute contact time, all 10 of our surfaces achieved at least 3 log reduction, including our dirt surface. So when we look at surfaces that were wiped with the chlorine instead of sprayed, we can see that there are fewer surfaces that achieved a greater than 3 log reduction for 0.2% and 1 minute of contact time. And we see quite a lot of variability across the surface types. When we let it sit for 10 minutes on the surface after having wiped, we did see an increase. So 7 out of 10 of our test combinations achieved greater than 3 log reduction. But again, we can see that we're still having some issues with our non-porous surfaces, wood through dirt, achieving the same disinfection. When we increased the concentration to 2% at either 1 or 10 minutes, we had 8 out of our 10 test combinations achieved greater than 3 LRV. And I'll note that you'll see that foam and dirt in particular, we did not achieve greater than 3 LRV when we wiped the surface with chlorine. What we were able to do was find that there's no statistical difference between the types of chlorine, so between NaOCl, HTH, and NADCC. And so we combined all of those results. So now this data is being presented as 0.2% in the yellow for 1 minute or 10 minutes of contact time, and 2% concentrations in blue for 1 or 10 minutes of contact time. When we looked at just surfaces that were sprayed, we can see that in total, 32 out of our 40 test combinations achieved greater than 4 log reduction. But less than half, only 17 out of 40, achieved greater than 4 log reduction when the surfaces were wiped. And spraying was associated with a significantly higher log reduction value than wiping. Now, if we dig into this data a little bit more and we look at our non-porous surfaces, we can see that surfaces that were sprayed out of all of the possible combinations of concentration and contact time, all 20 tests achieved greater than 4 LRV for non-porous surfaces that were sprayed, and 15 out of 20 for surfaces that were wiped. But we notice differences when we look at just the porous surfaces, and we'll see that a little more than half of all of our tested combinations achieved greater than 4 LRV on sprayed porous surfaces, and only 2 achieved greater than 4 LRV on porous surfaces that were wiped. Non-porous surfaces were associated with a higher LRV for both spraying and wiping. And so surface material is really critical in the disinfection. In conclusion, Vibrio cholera was reduced greater than 3 log on most surfaces when either sprayed or wiped with 0.2% or 2% chlorine and left for a contact time of 10 minutes. I will note that achieving this level of disinfection on packed dirt was challenging, and we actually only had 1 test combination spraying packed dirt with a 2% chlorine solution and allowing the contact time of 10 minutes was achieved this level of disinfection. We saw no significant differences between our chlorine types. Significantly higher log reductions on surfaces that were sprayed and significantly lower log reductions on porous surfaces. And in total for our recommendations, so we've developed this little table to be able to sort of identify, based on the surface material and the surface contamination, a recommendation for spraying or wiping. And you'll notice that for spraying, we recommend a 0.2% chlorine concentration when the surface contamination is either low or high and the surface material is non-porous or porous, as long as it's contacted for 10 minutes. But for wiping, we always recommend a higher chlorine concentration of 2.0%. For both porous and non-porous surfaces. And it's important to note that using a higher concentration, you would need to make sure that you have some sort of protection on your hands, such as gloves, when wiping the surface. Overall, though, our most important takeaway is that it's really critical to ensure that sufficient disinfectant is applied to the surface via either spraying or wiping, so that the surface is visibly wet, so that we know that we have enough chlorine that is in contact with the surface to ensure disinfection. And I want to make a note that we don't recommend to spray chlorine in households or healthcare facilities when people are present. And I want to also highlight that this research does not provide evidence on which interventions, either disinfection kits or household spraying, we should be implementing. So with that, I'd like to thank you for your time. Thank R2HC for funding this research, and I'll note that this was just published in Journal of Water and Health. Thank you.
Thank you, Gabrielle. Really appreciate your presentation. I'm gonna see if any questions come up in the chat. In the meantime, I just have a question about the porous versus non-porous surfaces. Why do you think there was such a big difference between kind of the effectiveness for those 2 different surface types?
So I think a couple of different reasons. So I think wiping chlorine across a porous surface, there's already a lower amount of chlorine that was in contact with the surface. And then especially for 1 minute contact time, less that intruded down through the pores into the surface material to disinfect prior to neutralization. And then also, if we think about the dirt surfaces, for instance, there's the potential for more organic load there, and less chlorine, again, getting on the surface. So there's less available chlorine for disinfection.
Gotcha, thank you.
Mm-hmm.
Let's see, last chance to ask a question in the chat box on Tappable. Oh, this always happens. There's always a little delay and then there's a bunch that come in. How did you measure the chlorine contents? What materials did you use?
Yep, so I prepared the chlorine solutions each day before testing. So I used stock HTH, NaOCl, and NADCC compounds. And then I used a Hawk ideometric titration kit in order to confirm the concentrations of the solutions were within 10% of our target, 0.2 and 2%.
Great, thank you.
Mm-hmm.
And there's a question, it says, was the spray a larger volume, I assume, of the chlorine?
Yeah, so the spray was, I think it was a milliliter more volume that I believe was in contact with the surface. So the towels that we used to wipe across the surface, those were soaked in chlorine until the towels could absorb no more chlorine. So we applied the same amount to each towelette, but there was always extra residual chlorine solution that would drip back into the Petri dish before we wiped it across the surface. So the towelettes were not dry by any means, but that is something that we weren't able to do, was quantify exactly what volume made it in contact with the surface via either method.
Thank you. And then we have time for one more question. Which method do you think would be better implemented in disinfection kits?
So spraying historically has been carried out by a trained agent external to the household, and so the wiping has been wiping or soaking a material is what is envisioned as the use case for a disinfection kit. I think what's really critical here is thinking about how do we ensure that chlorine is in contact with the surface. So if we are like using a cloth to wipe a dirt floor where there's been a spill, like a fecal spill or a vomiting spill in a household, that might not be enough chlorine applied to that surface. And so we might need to think about other strategies besides wiping in order to ensure that we have enough chlorine in contact with those surfaces. But I do think that what is interesting about this study is it shows that we really need to consider the surfaces when we're thinking about how to effectively disinfect them in field contexts.
Thank you for answering all those questions. If any more pop up, you can always respond in the chat tab. So if anyone thinks of questions, please do add them there. Thank you to both of our presenters for those great presentations and for some really engaging answers to questions. I just have a couple of announcements before we close out. Please do visit our poster session. It's our final poster session of the conference. It's this afternoon at 3:15 Eastern Daylight Time. And then after that, at 4:30, we have a trivia night, which should be kind of a fun way to close out the almost last day of the conference. And then the final plenary session is tomorrow morning. Thank you all for joining. I really appreciate you spending time with us, and I hope you have a great rest of your day and rest of the conference.